So, okay. And the talk, the title of the talk is Site-selective infrared spectroscopy to investigate protein conformations and dynamics. And I will stop sharing, and the floor is yours, Megan.
Okay, unmuted myself. Okay, we're all ready. I'm going to minimize this. Oh, shoot, I can't. Sorry, 1 second. I find having the entire half of my screen hidden by the pictures to be distracting. Okay. All right, thanks Thomas and Maxim for the introduction and the invitation to talk today. Yeah, as my title sort of said, we are after site-specific 2D IR spectroscopy with the goal of understanding the motions of proteins. So, because really the field of biochemistry has gone beyond the structure function paradigm, and we've now kind of realized that proteins, just like macroscopic machines, they're the machines, molecular machines of a cell, and so they really need to move in order to function. So anywhere from small scale changes to really dynamic molecular recognition. So this is kind of the aspect of protein function that we're after. And to do that, we've been developing site-specific 2D IR spectroscopy. And I'm just going to motivate why IR spectroscopy in particular. There's a lot of biophysical techniques. IR is not the most popular. But it has both high spatial and temporal resolution, which I think is really suitable for tackling the challenge of studying very complex systems like proteins. So obviously proteins, as I said, are highly dynamic rather than a structure. A modern view is energy landscape, where you have kind of a hierarchical structure anywhere from kind of slower motions, maybe domain changes. But then as you go to smaller scale structural changes, you can go to loops, side chains, and even coupled solvent. The structural changes become smaller, and hence they occur in faster timescales. And so, as I said, the community now really well accepts that dynamics are important to function. The role of these more rapid motions are more controversial, and I would argue because they're more challenging to measure. Hence, we're applying IR spectroscopy because it really has a very fast inherent timescale. We're assured to capture all of our states. I'll compare us to NMR, which is awesome, but that could be a challenge. So I saw an earlier talk, why are these motions important? Why could they even be? Well, if you're Josh Wander and Andrew Liu, who does NMR order parameters, you'd argue that in fact, the major contribution of entropy changes in protein molecular recognition involves just this rapid side chain motions. So if you want to understand the molecular mechanisms of thermodynamics, it could be important. Also allostery, a lot of proteins, you have domains and you might have a ligand bind one part of a protein and it's transmitted through very small local changes throughout that protein to impact a function on the other side of it. Again, small scale changes that could affect function. Where I've been interested in specificity, people like me think proteins are evolved not for affinity, but for specificity within the cellular milieu. How do you achieve that with a limited chemical space? Well, through tailoring dynamics and allowing proteins to adopt to other proteins or not. And finally, reorganization energy is an obvious example. Redox centers, it's well known when they're embedded into proteins, it restricts their motions, and unlike redox sensors and solvents, you see you don't have this rapid scale reorganization, and it basically makes electron transfer more efficient in protein systems. So some examples where protein dynamics and fast timescales could be important. So as I said, IR has a great inherent fast timescale. It also has a really awesome spatial resolution in principle. So again, proteins to a chemist are a really complex molecule, very large. And so just showing, polarity surface view, the contribution from one part of a protein to function could be very different from another part. So we really need a way to probe different parts of the proteins. IR is nice in that our chromophores can be as small as a bond in principle. So we can probe local regions. Of course, like this group knows, if you take an IR spectrum of a protein, it's highly congested. So although you can in principle get bond-specific resolution, in practice, it's not that easy because just so many overlapping absorptions, you can't see the absorption due to a single bond. Conventionally, the amide region here is most used for studying proteins with IR, but of course, that gives you secondary structure information, but it's more global. You can do some isotopic labeling, but by its nature, the amide bond is delocalized. So we've kind of avoided that. And so us and a handful of others have instead focused on the fact that there's a transparent frequency window in a protein IR spectrum where no native protein absorptions occur. So what you can do is put a bond that absorbs in this region, and you can therefore see a bond within the context, a single bond within the context of the entire protein, and get that spatial resolution that we're after. And also because we're choosing bonds, they tend to be more local mode rather than delocalized, so we can get bond-specific information. They're small, so compared to like a visible chromophore, they're minimally perturbative. And we're, again, local mode. We're choosing them to be very simple. So the physics of our probes are not of our interest. We want them to report on the protein. So really the philosophy of my group and others is that our protein systems are really complex. Let's try to simplify the measurement as much as possible. So I said a variety of people have been doing this. Really the first 2D IR experiments on proteins were focused on heme ligands, carbon monoxide and other small molecules. And that's in that transparent frequency window, but of course that would limit you to studying heme proteins and that one spot in a heme protein. From there, people use frequency-resolved probes with substrate analogs. These, of course, probe a very important part of the protein, the active site, but that again limits you to studying the active site. So to probe any part of any protein, people have been appending amino acid side chains with these frequency-resolved probes. And that, again, allows you to look at different parts of a protein. We want to have a global view of their dynamics and how it contributes to function. So of all these probes, I've been using Cyanophenylalanine since I've begun my group. We like it because that cyano group, of course, is in the transparent frequency window. This aromatic nitrile is, at least it's intense enough of a probe that we can do experiments at low millimolar concentrations of proteins. And I also like it because we can fairly easily incorporate this probe wherever we want in a protein via the amber codon suppressor technology developed by Pete Schultz a while ago. So all we have to do is take our gene sequence, put in what's normally a stop codon, and then co-express with synthetases and tRNAs that have been laboratory evolved to incorporate cyanophenylalanine. And the bug will then incorporate the residue wherever we direct it without any other modification to the protein. So I know that this audience should already kind of know 2D IR spectroscopy, so I'm not going to, this is my methodology slide. So just to get everybody on pace, we put a probe in a particular spot in a protein. The absorption spectrum would be characteristic of that particular environment. A single one would be kind of characteristic of a distinct state. And the whole range of substates of the environment around that probe, of course, would lead to inhomogeneous broadening. We can get at that inhomogeneous broadening and the dynamics within it, of course, through measuring time-dependent 2D IR spectra. Common analysis, of course, is the center line slope method that gives you approximation of the normalized inhomogeneous part of a frequency correlation function, which then you can describe it, say, like a simple Kubo model. But just the gist is, of course, we have an absorption band, and then by doing the 2D IR, we can get the dynamics that are describing the frequency dynamics within the band. And of course, that'll tell us something about the dynamics of the environment around the probe where we placed it. All right, so been a slow year. So today I decided we've been working on this, or we've been trying to do site-specific 2D IR to learn about protein molecular recognition for a while now. So I thought I'd take the time just to step back and make a summary. We've now done this for 3 different systems, all of which cases where dynamics have been evoked in molecular recognition. My time here, I'm gonna try to just kind of give you a brief survey of kind of the 3 systems we've looked at and how we've learned about how dynamics are impacted in molecular recognition. So the first system that we accomplished this was simplest system at circumology 3 domains. These are recognition modules that are very common in eukaryotic proteomes. They recognize a proline motif that's also very common. So our long-term goal is to understand how the dynamics might impact the specificity, because all these proteins have the same structures, same conserved residues at the interface, and could dynamics be involved? Moreover, dynamics are implicated in their recognition through entropy. These binding tends to be entropically unfavorable, despite the fact that it's a hydrophobic interaction that is presumably going to release a lot of water. So just motivating the problem here, I'm mostly just focusing on our site-specific study. So we incorporated the cyanophenylalanine individually at 3 different residues along the interface. These are normally conserved tyrosine residues found in almost every SH3 domain. Put one on a mobile loop, one at a conserved aromatic site in the core, and then another here for more of a control. So here are the linear spectra of each of the residue encolored in the absence of ligand in black when you bind its proline-rich ligand. Kind of the gist of this data is that it's not really exciting. The linear spectra aren't actually that sensitive to binding. A little bit of changes, but more so of these residues in the core, but they're really small. The largest is less than a 1.5 wave numbers. The dynamics were actually much more sensitive and differentiated the different residues. So probably see the residues away from the active site 21 and 5 here were basically not that sensitive. This residue at the core, we saw faster dynamics was quite perturbed upon binding. And then the 3 residues along the interface showed all different behavior. 11 here, despite the crystal structure showing a high engagement, we saw no effect of the dynamics. And then 13 to 57 here, 13, it got slower and an increased offset. 57 here, we actually saw faster dynamics. So this is a small system. And so this is where one case where we did try to understand the spectroscopy a little bit more. So we did do MD simulations of each individually labeled protein in the presence and absence of the ligand. And here I'm showing you the correlation function of the projection of the E field on the bond. And they, for some of them, they sort of match up. I mean, these got, we don't capture this offset that you see in computationally. 11, I guess, didn't change. 13 and 57 did recapitulate. We saw a large increase, slower dynamics, an increase in the offset. And the offset wasn't captured, but the faster dynamics upon binding the ligand was captured at 57. So the nitrile is a little bit hard, and this is like probably a problem for the field because it's also sensitive to hydrogen bonding. So we also looked at that. And I would say these residues here that weren't quite described as well by the E field did show, well, and the fact that they showed no change in hydrogen bonding, they match up. And then 25 here did show hydrogen bonding to protein backbone in the absence of ligand, but no hydrogen bonding upon binding the ligand, which just would explain this data. And then the hydrogen bonding with 13 and 57 kind of makes sense with 13, but 57 the opposite. So I guess maybe the point being, it is a little bit challenging to describe the spectroscopy, but it is sensitive to either hydrogen bonding or the local electric field. And then just looking at 57 and 13 here, why they do appear to be different. At 13, it's actually in a very charged region of the binding surface. So upon binding, all of these charges that he sees probably become restricted, leading to this offset. In comparison, 57 just sees water in the absence of ligand, and then upon binding, it goes into a pocket here and points directly at a carbonyl group. And basically the dynamics are just due to the wiggling fluctuations between these groups. So kind of give you a molecular picture. But the gist of this all though is we were able to, for the first time, show site-specific dynamics throughout a protein and see how it responds to molecular recognition. Really, these 2 residues here and the core were more sensitive, although they appeared to be sensitive for different reasons. Whereas 11 here, again, showing that the crystal structure could be misleading. We does not seem to be as much engaged. All right, so the next example I will tell you about is our studies of electron transfer proteins. And in particular, we use that as a model system, plasticine and cytochrome F, which are 2 members of the photosynthetic apparatus. Why these are of interest is because they're thought to have very dynamic molecular recognition. In fact, what I'm showing you here is just a model derived from NMR, not conventional NMR, but just paramagnetic relaxation experiment. So you can kind of map out what parts of plasticine at some point get near the paramagnetic heme center. Or you can look at chemical shift perturbations. But I want to point out, look at the values here. They're tiny. And the reason that is because it's thought that the majority of the population is actually adopts a loosely bound state, or they sometimes call it a counter complex. And if it does form a tightly bound state, it's in rapid equilibrium on an NMR timescale. So a case where using a technique with a faster inherent timescale would be useful. So we looked at this. It was less amenable to labeling, but we were able to put 3 of our cyanophenylalanine probes. One question in the field is just the head, or does the side also engage in this interaction? So we put one here, 36, clearly in kind of this hydrophobic head, 188 here, which is in kind of the side polar patch, and then 90 here, which is kind of in between, still kind of at the head. So here are the linear spectra. They were more sensitive than the SH3 example. And again, in color-coded to the residue is the data in the absence of cytochrome F, and then upon binding, the black data. So at all residues, more so at 36 and 90 than 88, we see a red shift upon binding, which should be reflective of more non-polar, less polar environment, and a broadening of all the bands. We did then characterize all of them with 2D IR spectroscopy. And as you can see, the dynamics were also very sensitive, more so at 36 and 90 than 88, again, pointing to greater contact or engagement at the head than perhaps the side of the protein. But note that the major feature we saw was upon binding, we saw a really large increase in the initial offset here, a little bit also the case at 88. Combined with, as I said before, we have an increase in the lined width. The data basically said that we see a large increase in inhomogeneity or heterogeneity of the environments of these residues upon binding. But what was interesting to us is that although we see a large increase in this heterogeneity, note in all the cases, our window is fairly small, the frequency dynamics are pretty much decaying by about 5 picoseconds. So that inhomogeneity is very rapidly sampled. So just to kind of quantify this a little bit more, fitting it to a simple model where we're separating the inhomogeneity into amplitude associated with really rapid dynamics, about 1 to 2 picoseconds, which is kind of characteristic of water dynamics, and an amplitude associated with slow dynamics. And I'm showing you the change in bar graph form here. So you can kind of see in all cases, the major effect is this faster dynamics, and it's about 3-fold greater than the amplitude associated with slow dynamics. So point being very fast. So now if I get kind of hand wavy and I say, okay, let's associate this increased amplitude with fast dynamics with a loosely bound state, and this slower dynamics with a tightly bound state, I can kind of estimate maybe what those populations are. So we get about 75% estimated in this loosely bound encounter complex, only about 25 in the tightly bound. I know that's a very hand wavy, but at least we're getting somewhere to quantify what these states are like. And the gist being is that, yes, these complexes are forming very loosely bound states. They're not tightly bound like a canonical molecular recognition of proteins. Okay, and finally, my last example is we've been looking at cytochrome P450s. It's a superfamily of enzymes. They do very challenging reactions. They can use molecular oxygen to incorporate an oxygen atom at an unactivated carbon center. So we care about these as humans because they're the major drug metabolizing enzymes. And the particular metabolites that are produced by these enzymes, where that hydroxyl group will determine drug efficacy and toxicity. So there's a motivation to understand or be able to predict the reactivity, which we cannot do yet. So I'll tell you today, we worked with these a little bit about our study of this archetypical cytochrome P450, because we want to know what's the role of dynamics in controlling the regioselectivity. So stepping back and this asking what could control the regioselectivity, I'm showing you the catalytic cycle, but honing in on this product determining step here. This enzyme creates a very reactive iron oxo species called compound 1, and it's basically a warhead. It can rip off a hydrogen from an alkyl carbon, and that leads to a rebound reaction. So basically wherever this hydrogen comes off is where you're going to get hydroxylation. So what's going to control the regioselectivity of these enzymes is either the intrinsic chemistry of the substrate, how hard it is to pull a hydrogen off of the different carbon centers, or as we argue for this next example, how the protein actually recognizes the substrates, holds it with respect to this reactive species, and lets one or another of those carbon centers even approach it. Okay, so we were after this question, can dynamics influence selectivity? So we really kind of set the system up for ourselves, meaning we're choosing it's probably one of the most well-studied enzymes out there. So there was a lot of previous data. So we had known that from literature that this enzyme will act on 2 different substrates. When it acts on its native substrate camphor, you get 100% of one product. When it acts on its another substrate, norcamphor, you only get about half of the 5-hydroxy product that's produced from camphor, about half of the 6-hydroxy, and then a little bit of 3. So I said this has been well studied. So the calculations have looked at the reactivity of the different carbon centers, and it does not explain this difference in reactivity. They basically are the same, which is why it's hard to direct chemistry at hydrocarbons. It's also not structure. There are high resolution structures of this enzyme with both its native substrate camphor as well as norcamphor. They're basically identical. The only difference is that the norcamphor has greater R factors. And so if it's not chemistry, it's not structure, it kind of has to be dynamics. It's kind of why we set it up. So this is what we were then asking, how do the dynamics of the recognition of camphor and norcamphor differ in a way that might explain this difference in activity? So I will point out that there had been known for about a decade that this enzyme does undergo a conformational change upon binding its native substrate camphor. In the absence of substrate, it goes from kind of an open state involving a helix, loop helix, FG helix here. And upon binding the substrate, this motif basically closes down and clamps on the active site. But it had been not studied whether or not the same conformational change occurs upon norcamphor. And so that's kind of part of the things that we were after here. So again, we're using our cyanophenylalanine probes, and we incorporated them at 5 different residues, several of which are either on or pointed towards this FG helix to be sensitive to that. 196 here, which is pointing directly at the carbonyl group of the substrate. 305 is kind of a control, and then I'll just bring up a little bit of CO data at the end. So here we're looking at the protein in the absence and the presence of each of these different substrates. So here are the linear spectra. Throughout this part, the free is in black, the camphor is in blue, and the norcamphor is in green. These residues, unlike the ones I've showed you for the last 2 proteins, which the linear spectra were kind of boring, or the differences, these ones actually showed different changes, and they were all kind of different at every residue. So I don't want to be too tedious, but I'm going to try to go through each of these residues fairly rapidly, and then kind of bring it together to tell us about the kind of a summarized model of the recognition of these 2 substrates. So first, just looking at these 201 and 305, which are farthest from the active site, they were less interesting, so we're not going to spend that much time on them. They were sensitive to binding substrate reproducibly, but not as dramatic. And the one thing I just want to point out is that 201 here was fairly asymmetric and did in fact fit to 2 bands. And I'll bring that up in a moment here. But really focusing, due to limited time, on the 3 residues that showed most interesting changes that are kind of closest to where the substrate binds. So first, looking at 98, this residue is pointed up towards this FG helix, although of the 3 farthest from the substrate. So showing dynamics weren't as interesting, but the linear spectra were. The free and the camphor looked like nice Gaussian bands, basically superimposable. Upon binding only camphor, we saw a shift in the absorption. We also, my student came to me and said, it best fits to 2 Gaussian bands. I kind of looked at this, maybe not. How we decide whether there are multiple bands, well, you ask what best fits, but you can go and do second derivatives, and we do pull out 2 minimum, supportive of 2 underlying states. And this is another reason why 2DIR can be useful for us. In addition to getting dynamics, we can take advantage of the fact that the 2D, of course, depends on your transition dipole to the 4th power, whereas the linear depends on the square. So if I have 2 underlying bands and they have different transition dipole strengths, they will differentially appear in the 2D versus the linear. And so here I've taken the 2D diagonals of the 3, the data in the 3 bands and overlap them to accentuate the line width changes. And you can probably see that the camphor clearly has asymmetric shoulder. And it did in fact best fit to 2 states. And this was a little bit non-intuitive when we first got this data, because as I mentioned, upon binding substrate, the camphor, it goes from an open state to a closed state that's thought to be more conformationally restricted. And here we're going from what looks like 1 state to 2 upon binding. However, what's interesting is that this residue, although it does seem to have heterogeneity in the bound state, it now looks exactly like the fit that we got for 201, which is right next to it in space. So although they retain heterogeneity upon forming this closed state, these 2 residues now kind of see the same environment. Okay, and then pointing out a NOR camphor basically had no effect. The fact that NOR camphor is not the same is a theme of this section. All right, so, the second residue, which I guess this will be the 4th in our thing that we looked at, was 87. This residue is expected to also be sensitive to this helix loop helix thing motif, but it is could also be sensitive to substrate. So I'm showing you the linear spectra and the 2D diagonals. Again, there are clearly multiple bands going on with the free and the NOR camphor. The free is broader, has the shoulder. Upon binding camphor, you have this narrow Gaussian band. Also shown here at the 2D diagonals. You can probably tell, obviously, from the camphor complex, it follows the NOR camphor complex follows the camphor complex on the lower frequency edge, but has this shoulder, and it's quite apparent, this extra bump here in the 2D diagonals, that the NOR camphor is 2 states. So, so 2 states for the free, so it definitely well fits to 2 states here. And then fitting the bands for camphor and NOR camphor. The camphor was a nice Gaussian band, suggesting in the closed state you populate 100% one conformation. NOR camphor, it did well fit if we assumed that one of the bands was the exact same frequency in line with as we found in the camphor complex, so such that it populates 1 state that's the same, and then pulled out fit to a second additional band that appeared at slightly higher frequency. But the areas are about 50-50. So this suggests in the NOR camphor complex, you're populating about 50% of the closed-like state, but also populating another state. So I'm just showing you back the 2DIR spectra here, just showing, although we do have these underlying multiple bands, they're so overlapped that we did not initially try to account for both of them when we analyze the 2D spectra. So when we do the center line slope, we are doing it over the max of the entire band. I point out, because of that, any interconversion or dynamics amongst these multiple states are going to contribute to our spectral diffusion. So here are decay curves that we get, and for both the NOR camphor and the free, we saw a greater offset or slower dynamics, suggesting that these 2 states slowly interconvert, which would be consistent with sensitivity to this larger scale structural change in this helix loop helix. And I do want to point out, just like the linear spectra, because we have additional data, if we assume now that this camphor complex or the NOR camphor is adopting a state like the camphor complex, we can then fit the 2D data, assuming a camphor FFCF and another state with a different one, and we can well fit it. We extract out this red curve. So the 2D data was also consistent with a superposition of 2 states that slowly interconvert in the case of the NOR camphor complex. All right, our RAST residue is 96 here. And this is interesting because this cyano group is pointing right at the carbonyl of the substrate. So it's probably going to be sensitive to its motion. In this case, we saw differences in our perturbation in the spectra upon binding both substrates. You look at the camphor and the free, it looks like there's not a large change, just a small shift, but note that the line shapes are actually quite different, and you can probably see that more here with the 2D diagonals. In fact, the free was best fit by 2 bands like we found for the other residues, and that's supported by second derivative. We don't pull out 2 minima, but the minimum of our second derivative does not overlap with the max of our linear absorption as it should if it was a single state. So the case of the camphor, though, it's again a nice single Gaussian band upon in that complex, suggesting it's seeing 100% of this closed state. NOR camphor, you can probably see it has this side feature here, best fits to 2 bands. Although in this case, we could not fix this the second band to be equivalent to the camphor clays. It did not fit, neither the linear nor the 2D data could be fit as a superposition. So these two do appear to be kind of two unique states in the Nor-camphor complex, suggesting that this is consistent with the fact that this residue probably is sensitive to the substrate itself, and they are different. Again, pointing out that we are doing the analysis over the entire band. So the frequency dynamics between these two bands would contribute to our spectral diffusion. However, at this residue, unlike 87, we actually found faster dynamics in the free and the Nor-camphor, despite the fact that we appear to have two underlying distinct states where the camphor appears to be one single state. That must mean that these states are rapidly interconverting. So again, Nor-camphor, we're kind of populating a 50-50 state that are rapidly interconverting. In the case of the free, we kind of think this is probably just due to hydrogen bonding with water. In the open active site, it's water filled, so that would make sense. There isn't water in the active site of the Nor-camphor structure, but it could be a possibility, but we think probably a better interpretation, or an alternate one, is that you actually have rapid rotation of the substrate in the pocket, and this is consistent with previous MD. And that sort of rotation would, I think my model, allow multiple carbon centers to approach. I'll get that model in a second. If you look at, we have another probe, a CO that we looked at earlier that is also should be sensitive to the substrate's motions. It's kind of on the other side of the substrate as 96. And just like 96, if we compare the dynamics with this probe in the camphor and the Nor-camphor complex, we see faster dynamics when you're bound to this Nor-camphor substrate. Okay, so just kind of putting this together, we looked at a variety of probes, many of which were directed towards this FG helix, and we anticipated would be sensitive to that. At all residues, to a certain extent, we saw a change in the spectra upon binding camphor that was not recapitulated upon binding Nor-camphor. And these were associated with fairly slow dynamics, suggesting that upon binding Nor-camphor, you don't get the same conformational change to that same closed state that you get upon binding its native substrate camphor. But at the same time, in the active site, we saw more rapid motions in the case of Nor-camphor for the two probes, the CN96 here and the CO, which are directly contacting the substrate, suggesting that while you have this disruption and this larger scale structural change, it leads to kind of a loosely fit or a wiggling substrate in the pocket. And we can come up with a mechanism that then explains the catalytic outcome, that basically we have rapid rotational kind of few picosecond motions of the substrate that allows both the 5 and 6 to approach this reactive center, which is very rapid. And although this guy, this compound 1 is very short-lived, he would be longer lived than the rotational picosecond motion. So we kind of have a Kern-Hammett sort of mechanism where we saw 50-50 populations. So these energy levels would be at the same spot, and they'd have about equal free energy of activation then to producing about equal of the 5-hydroxy and 6-hydroxy products. On the end, showing though that fast dynamics could contribute to the outcome of catalysis. So with that, I hopefully showed you how we've been able to get employee site-specific 2D-IR spectroscopy to get a kind of a global read of how dynamics are impacted by molecular recognition and the context of 3 different systems. That I have a lot of people I could thank. I just want to point out mostly Sashari Ramos here. She is left me. She's now a postdoc with Martina Havenith in Bochum, Germany. Pretty much all of the cyano 2D spectra that I showed you was collected by her, a little bit by Amanda Lasur. Ed Bassom collected the CO data, and then we showed you a lot of labeled proteins. I'll call out Claire Mamisser and Rachel Hornis and Amanda Lasur for producing those, but I also have to call out, these are just a few of the undergrads that, that support my group. And I guess this DOE, NIH, and NSF funded the different projects. So thank you.
Thank you very much. So the talk is open for questions. There is an anonymous attendee who asked a question. Maybe you already addressed that later in your talk, but the question was, what does the offset in the correlation function depict?
Yeah, so it's our window is limited by the lifetime of our probe, unfortunately. So we're, we measured out to like 5 picoseconds typically. So that's just basically increased inhomogeneity that is interconverting slower than our short time window. So could be anything, 10 picoseconds and longer. We unfortunately can't tell you.
Chris, you have a question.
Yeah, Megan, really beautiful talk. Thank you. My question is focusing on the plastocyan and cytochrome F complexes. As you note, right, the hard part isn't, in some sense, getting the data. We spend most of our time on that. It's trying to figure out what all of the numbers mean in the end and giving some physical interpretation. And so you offered one possibility. I want to offer another and get your feedback. Right, you've got this loose complex and all of your probes point into the space in between the two proteins. There's almost certainly water in the space between the two proteins, but it certainly doesn't behave like bulk water, right? It's confined water at that point. Everything should slow down a lot.
Right.
Your interpretation was 1.2 picoseconds, that's water. That must be the stuff from the bound complex. But if I remember correctly, from that manuscript, the other thing that happens, not only do you see increased values for delta, but you see decreases in the motionally narrowed contribution. So I want to offer the alternative that maybe the stuff you see that decays fast with less motional narrowing is the tightly bound complex, and the loosely bound complex is the one where you don't have the 1.2 so much of the 1.2 picoseconds, right? You should get lots of 1.2 picoseconds where you have lots of bulk water, and you should get different behavior when you have the loosely bound complex. What if it's the other way?
That's a good point. I mean, I still don't understand the 1.2 picoseconds. To me, that's just kind of weird because it is so fast. But yeah, I get your point, and I guess it'd be another way to think about it. And I guess it's something to think about. But you are right. I guess I often, when I give a full talk, I'll put this with my carbon deuterium probes that we've put at the Medical Center. Right. And in fact, yeah, when we do, like, we see the shifts in our carbon deuterium probes and do like a Stark effect hand wavy argument, it goes from like a dielectric fourfold lower. So it's almost like isopropanol. So you're right, it is not bulk water in any way. And that is, I am confused why, I mean, I've set the data that are in a way contradictory. Yeah. I have started a collaboration. So Dmitry Maximov, excuse me, at University of Arizona says he's been doing calculations on these for a while. So I've put him on the task to try to help me understand this. It is such a big complex and there's metals involved. Hard. I don't want to touch it, but yeah,
it's tough.
But yeah, thanks for the suggestion. I think I'll need a little bit of time to digest that. But.
Fair enough. Fair enough. Beautiful stuff. Really lots of lovely data. Thank you.
Thank you.
Other questions? Otherwise, I have to warn Maxime. I will have to ask a question. Oh, Rika has a question.
Hi, this is it was an amazing talk. Thank you very much. I just I would have lots of questions, but a brief follow-up question to the vibrational lifetime question. You had this nice paper with the cyanide, right? How is that as a probe? Have you remeasured a few things where you saw this offset with the cyanide, maybe?
Yeah, it's oxygen, so we can make the probe, and if we keep it out of the light and in anaerobic conditions, it's great. As soon as we tried to put it, we did try to put it in a protein, just threw it in amber, threw it in the media, and the mass was off. So we didn't figure out what exactly is happening to it, but it does not survive a cell. So I really want to do cyanoselenocysteine, and I have a synthetic route in my head. Maybe I'll go do it myself this summer 3 times. But yeah, unfortunately, I keep thinking maybe we could do anaerobic expression, but that adds another layer of complexity to do expression. So I haven't gone there with limited resources. But
okay, so this answers the question why you didn't show it, because I always thought that this approach would have been very cool.
Yeah.
Can I ask a question? So when you introduce these labels, yeah, so this phenylalanine analog is fairly big, right? So are you sure that you don't disturb the function of the proteins?
Yeah, well, the cyano is not. So at every residue that I showed you, except for the plastocyanin, it is an evolved beast, a little beast. We couldn't be conservative. The sequence is so conserved that we did have to make some mutations. There's one where we put a glue, but the rest of the P450 and the SH3 domains, those are all tyrosine residues or phenylalanine residues. So the change in the chemistry is actually fairly minor. But we do characterize, we use circular diachrosm, and these are all molecular recognitions. So we do binding affinity assays, depending on the protein, whatever other biophysical techniques that we can do to support it. So these all are folded. Their molecular recognition, depending, system isn't dramatically affected, meaning it's not so dramatically affected that we can't push it to completely bound. The data is all in my SI or in the papers. You do what you best you can. If you report if they are perturbed. All of these were folded. They bind their recognition partners reasonably well. So yeah, there could be perturbation, and report that. If there was we've had ones where they don't fold or something, and we just kind of have to toss the data or not look at those residues.
Okay. Other questions? Well, if not, then I think we can thank Megan again. And